My name is Christina Manier, and today is Monday, April 8th, 2024. I'm going to be talking about Staphylococcus epidermidis and keratinocytes, but specifically keratinocytes that are dead and flattened, and those are called corneocytes. So first things first, Staphylococcus epidermidis is one staphylococci bacterium that lives on the skin. It's often referred to as a golden cluster of grapes because it takes a golden color in a petri dish, and when it multiplies, it sort of forms a grape bundle cluster. It's gram positive, so it turns purple when the gram stain is applied. It's catalase positive, so it produces an enzyme that breaks down hydrogen peroxide and turns it into water and oxygen. That's why we see the foam when you apply, sometimes when you apply hydrogen peroxide. So moving on to the skin, the epidermis. So this is the first layer of the epidermis. So there's different layers of the skin, and then of course the epidermis is multi-layered. This first layer attaches itself to the dermis using the hemidesmosome, and then throughout the structure, these cells attach to each other using desmosomes. These are basically fresh new cells, and they have all the typical organelles that a eukaryotic cell would have. Just kind of taking a zoomed out look, so the base layer is the stratum basale, and it's like a hexagonal column shape. You can see that it has the nucleus and the nucleolus still, and then as it moves through this process of terminal differentiation, it basically produces a bunch of keratin and excretes these lamellar lipid granules. Eventually the nucleus dissolves, and these cells at the stratum corneum layer, they have no nucleus. They don't have the typical organelles that most eukaryotic cells have, and they are dead and are going to just slough off eventually. Relative size, just a quick scale check. So the bacterium can be anywhere between like 0.2 and 2 micrometers. And then the corneocyte cell, when it goes from being a keratinocyte to the corneocyte, it's completely flattened, basically. And then so it can be anywhere between 30 to 50 micrometers in length, and then up to 1 micrometer in depth, width, height. Yeah. So this is the stratum corneum. It can be anywhere between 15 to 30 layers of corneocyte cells. They're attached with desmosomes, and they're wrapped in the cornified lipid layer, and then inside of them is a bunch of keratin, basically. On the surface there is the bacterium, and the bacterium is the bacteria is surrounded by the biofilm, which is one of the things that allows the bacteria to adhere so well to not just dead skin, but like a knife or a needle, or any foreign body really that you put that, like, I'm just thinking of, like an artificial joint. But this question of like how and why do Staphylococcus bacteria, how is it able to like live so well on certain surfaces? This is still a question that's being asked by a lot of researchers. So there was one study that I found that in 2021 it was published, and these researchers were able to figure out exactly like which protein was functioning the best in attaching the bacteria to these dead skin cells. So basically, they found out that these dead skin cells have, like, the surface still has a variety of different like glycoproteins and proteoglycans and glycolipids that are like, they just offer a variety of different binding sites. And then for the bacterium itself, they found out that it relies heavily on the domain A in the accumulation associated protein. So that's what I have illustrated there. That's the AAP, the accumulation associated protein. So while I haven't really been able to find super duper specific, like a super duper specific diagram of the corneocyte itself, it probably because of the lack of research, we're still kind of discovering this stuff. I was able to find a study that talked specifically about like which proteins are attaching. A note about my diagram also, I know it says corneodesmodome there, pointing to the corneodesmosome. I'm sorry for the spelling. I know it's corneodesmosome. So that's what I've got.